Look closely at this macroscopic footage of an octopus resting on a coral reef. The animal's spike-covered skin blends into the jagged rock, swaying slightly. This is tactile invisibility, the ability to match the precise three-dimensional topography of an environment in an instant. This presents a biomechanical paradox. A completely soft-bodied organism, lacking any internal bones, is somehow locking its tissue into load-bearing, rigid spikes. To understand how a creature creates structure from nothing but soft tissue, we must move past the macroscopic view and examine the surface of the skin. Cephalopod camouflage is an architectural problem. It requires the animal to generate physical, structural rigidity on demand. The specialized biological units responsible for these architectural transformations are called papillae. These networks are distributed by the thousands across the mantle of the octopus. When the animal is swimming or at rest, the epithelial layers of the skin remain flat and smooth. Upon activation, however, that smooth skin transitions into an elevated, rigid spike. These structures bypass the need for a permanent skeleton. Instead, they rely on motor neuron-driven muscular hydrostats to generate force. By dissecting the physics of the muscle tissue and the neural circuitry that triggers it, we can map exactly how this change occurs. The papillae function as a localized, deployable structural response, one that materializes only when the environment requires it. The muscular hydrostat is the mechanical principle that allows soft tissue to generate its own structure. The core axiom of this mechanic is that while the tissue can change shape, it maintains a strictly constant volume. This diagram shows the physics in action. Muscle tissue is mostly incompressible water. Volume squeezed on the x-axis must expand on the y-axis. Unlike fluid-filled skeletons, cephalopod tissue is solid. Three-dimensional fiber arrays pack together, providing structural support to resist pressure. We can apply this mechanical model directly to the biological structure of a papilla in its relaxed state. This system allows for rapid shape changes. Because there are no heavy bones to move, the transition is almost instantaneous. The muscular hydrostat principle turns the fluid nature of the octopus into a physical asset, using pressure to create form. By looking inside a single papilla, we can see the specific arrangement of muscles required to build a spike. Concentric muscle fibers are arranged in a circle around the base of the structure. This cross-section highlights concentric fibers at the base and vertical longitudinal fibers in the core. To begin, concentric fibers contract aggressively, squeezing inward. Since volume cannot change, that squeeze forces internal mass upward, creating lift for the spike. Simultaneously, the longitudinal fibers tense. This limits the upward extrusion, shaping the tissue into a sharp, jagged point rather than a rounded bulb. This dual contraction sequence is incredibly fast, allowing the rigid texture to appear in just a fraction of a second. The rigid spike is the result of these competing muscle contractions, fighting against the incompressible fluid within the tissue. Controlling this physical interaction requires a complex map of motor neurons. Neural signals travel from the central ganglia directly to the dermal layers of the skin. These neural pathways terminate directly in the erector muscles of the papilla, providing a dedicated control line for each spike. High-frequency firing initiates the initial thrust, but the system must then maintain that position. To hold the spike in place, the octopus uses a locking mechanism, a sustained neural signal that keeps the competing muscle groups in a state of rigid tension. This locked state gives the skin enough structural integrity to resist being flattened by water currents. It also ensures the spikes remain rigid when the octopus is pressed against the solid surface of the reef. Precise neural mapping is the framework that allows fluid tissue to function as a temporary, load-bearing skeleton. When we zoom back out to the entire mantle of the octopus, we see the scale of this mechanical effort. Thousands of individual papillae are firing and locking simultaneously to create the illusion of a solid rock. This video shows the skin erupting into texture. Maintaining this state requires constant neural activity and significant metabolic energy. This active architectural strategy is distinct from the passive shapes of other marine life. It is a continuous, powered performance. The causal sequence is clear. Motor neurons fire, the hydrostats contract, and the papillae lock. By using these neuron-locked spikes, the octopus bypasses the need for bone, reshaping its body to match its environment. Mimicking coral is more than a visual trick. It is a feat of biomechanical engineering. To master a three-dimensional environment, the octopus must physically manipulate its form. Camouflage here is a matter of active body topography. This demonstrates that a complex nervous system, when paired with specialized muscle arrays, can take over the role of a skeleton. These biological principles provide a potential roadmap for engineers who want to design rigid, deployable structures using only soft, flexible materials. While the term neuromuscular sorcery is often used to describe this effect, our mapping shows it is a grounded reality of physics and biology. Ultimately, tactile invisibility is the direct result of a highly advanced nervous system operating as a dynamic, programmable skeleton.